very unusual observations that have been discovered in 2018, which means all of the fact checkers that have been saying, no, it's impossible to convert back from mRNA into DNA. Well, they should have known better before they ever said that, because the information of how our potentially natural cells might be using these selfish genes, that's been already around since 2018. So first, let me give you background on this selfish gene. So how does it work? Normally, when a gene exists inside our DNA, in order for the gene to be used, it has to be copied into RNA. Then RNA will exit the nucleus and it's used as a template or a blueprint in order to produce a protein. And a protein, think of protein as a tiny molecular robot that will do all the work inside our cells. So that's how I think of robots. Robots basically make the cell function. You need the blueprint to build the robot. That blueprint is the RNA. But you don't go backwards. So you cannot go from protein to RNA. And normally you do not go back from RNA to DNA, unless you were to have a very specific protein, tiny little molecular robot that could do it. It does exist in nature. It's called reverse transcriptase. So as an example, HIV patients, they have that because HIV, the virus, produces one of these. It has a template to produce reverse transcriptase, and this is how HIV can insert itself into our genomes. And this is also why HIV patients, amongst the cocktail of medications that they have to take, one of the medications they have to take is a reverse transcriptase inhibitor. So it's an inhibitor that will stop that protein that can reverse transcribe RNA back into DNA from properly functioning. Now, we humans normally do not have it, except this one gene, the selfish gene, the line 1 gene. So how does it work? That gene is copied into RNA. The RNA is then exits the nucleus, and inside our cell, the environment outside the nucleus, inside the cell, it's called cytoplasm. Inside the cell, the RNA is used as a template to produce the protein robot. And here's the unusual part. This protein robot is selfish in that it doesn't go and does anything for the benefit of the cell. It goes back, grabs its own RNA, because remember, there will be plenty of RNA produced. It will grab its own RNA and sneak back into the nucleus. Now, one of the components of this protein robot, it has the ability to cleave the DNA. It will just cut one of the DNA strands and then use the RNA as a template to reverse transcribe it back into the DNA, and while inserting that new DNA right into our genome. So it's very similar to how CRISPR-Cas9 system works, if you're familiar with that system. So it's almost like we have our own version, and this is how this selfish gene can go back and read itself into our genomes over and over and over, many times over. So you can see if you dysregulate its function, how it could be promoting potentially something like cancer growth, because you could be really distorting proper genetic material. So in the context of these specific selfish genes, apparently once in a while, extremely rarely, it could supposedly grab other mRNA, not its own, and bring it back into DNA. But that was assumed basically as impossible or so extremely rare that it's negligible. But I'll tell you right away, after we go through the publication, that perhaps we should have known better.